All right, and we are live. I'm here with Mr. Matthew Gates, the man, the myth, the legend. Um, it's been a while since we had like a kind of a guest on FCP. Unfortunately, we were briefly taken down FCP 1 and 2 for a while. YouTube, they're giving some strikes on cannabis videos. Just the algorithm is really crazy. But we have some reserve channels. So um, I've been a fan of Matthews for quite a while. I recently joined social media on LinkedIn, and like whenever he posts, like I just learned so much. And I was like, "Hey, you want to just kind of come on and give a brief overview of like your work, stuff you're involved with?" And I am having like my class at Wichita State Watch, and Matthew is like the perfect guy. So Matthew, if you can give you or if you can give us a brief introduction to yourself. I know a lot of people already know who you are, but for those who don't.
No, it's a niche subject that I cover, so I'm always happy to do that. My name is Matthew Gates. I'm an integrated pest management specialist, and I've been doing that kind of work for about 17 years now. And that basically entails supporting growers, not just in cannabis. I've worked in other crops too. Like I've spent 2 years in China, I often mention, which you'll probably see some of that influence here in this presentation. And I worked in sorghum and tea, but also cut flowers like Gerber, lilies, roses, and some vegetable crops too, and a bunch of other stuff, sort of auxiliarily in that way. So yeah, I have a ton of experience, a breadth of experiences, and I like to focus on plant health dynamics and also kind of understanding what what makes a pest really difficult to deal with, and something that goes maybe further than the surface level so that people can make informed decisions at like a really high level, even if you're just an individual grower.
I know if I've learned, I know I've learned a lot for you. Just like whenever I've been around the plant since I was a kid, and just like I always keep learning something. Especially around pests, because there's always something new coming. Like alternaria, we assume it's like septoria. Not exactly, So like stuff like that's popping up. But Matthew, I know you got a presentation for us today to drop a 70 slide PowerPoint to drop a bunch of knowledge. Crash
Don't
course.
scare them off like that. Don't scare them off like that. It'll go quickly, actually, because people are smart. And the really cool thing about video presentations is that you can pause it and then look at the slide longer than you need, and then or come back to it too. So that's why I like to really give a lot out there, a lot of visual aid, so that people can refer to it later. So yeah, but we're going to go over pests, and we're going to go over some example pests that you get in cannabis and what makes them tick, basically. So I won't go into the nitty gritty, but I will go over some aspects of their, development that makes them kind of difficult. Because if you've had difficulty with these pests, I just hear it all the time. People are like, "Oh, you have trouble with this pest. Oh, you must be such a bad grower. Oh, you don't take care of your plants. They don't get good nutrients." Or and I just think that's inaccurate and also unnecessary. It's not really the case. These things have been doing this for like hundreds of millions of years. They're pretty good at what they do, and we're helping them get better at it, actually. So don't be too hard on yourself.
Don't bug out.
Don't bug out. Yes. So is the presentation live? I can't see my own presentation.
Oh, see, let me add it to the stage here. There we go. And I can just go ahead and make this. Are you able to control it from your end?
Yes, sir. Is it enlarged?
Yep, on my end.
Okay, cool. All right, so the title for this presentation is Spring 2026 IPM Analysis, Pest Adaptation Trends for Cannabis. A little bit about myself. I already mentioned my IPM background. You can find more media about pest management in cannabis and other crops on my YouTube channel, Zenthanol, and also on X and Instagram at SyncAngel. You can also check me out on my site. There's a QR code in the upper right. And I have some selection of publications and symposia on the bottom left here. For example, I collaborated with a really good team of people at Cornell, and we made the New York State Cannabis Sativa Guidebook in Spanish and English, so you can check that out if you're inclined. And I'm also working with a cool team of people looking at a systematic review of neem. and its use in cannabis, and how how we know from other crops and other plants and other applications, what might make sense to do and what doesn't make sense to do. So you can look out for that, hopefully publishing very soon. But I've also talked about things like viruses in cannabis and various different kinds of taxa, various mites and insects and things in cannabis. I also was even interviewed by Japan, a Japanese broadcast, which is pretty cool. So I'm not actually a Magic player much, but I hear that it's popular. So I decided to make this little image on the right. Maybe some people understand the joke. But basically, I like to use this concept called the Everswarm when talking about pests, because it basically helps us acknowledge that pests are constantly evolving, and we're not dealing with just one kind of pest, we're dealing with many kinds of pests, and each species has a bunch of subpopulations. In addition to that, the environment changes quite a bit, and so pests are also adapting under the stresses of a pretty cruel environment, at least in some places. Also, the agricultural, chemical pesticides and other sorts of things, even biocontrols that are being used. These are all causing selection pressures. Even physical controls, to a large extent, has an effect. It's just hard to kind of tell in the field. So there's many cards to play, which is the joke here, and many contexts to consider. I would say that a robust IPM strategy, whether you're an individual home grower or a commercial entity, is to have both proactive and reactive plans that can include natural and supplemental biocontrols. It can include what's becoming really popular is molecular priming or genetic priming using microbes or compounds or things like this derived from plants or other organisms to kind of sort of curve the development of a plant, or perhaps even just prime the plant if you know certain pests are going to come around. But there are some disadvantages to that, which we'll go over. But also, I want to say that it's also incredibly common, and I'd be remiss to not mention that sometimes, especially in places where you have leadership, so like a commercial setting, there can be inflexible perspectives that force you to conform to expectations that may not be reflective of reality, which is just a lot of words for saying that you can amplify confirmation bias. You might think that because your plants are grown a certain way or in a certain environment that they can't be threatened by pests, that pests are too good for them or the plants are too good for them, but that's not really the case, and it leaves you vulnerable to a lot of issues. So you want to, maybe if you're in a leadership position, don't be afraid to accept or understand that some of these things are happening, and it might not be the fault of your crop scout or whoever is giving you the information. You have to move forward with that information, hopefully make good decisions if they're practicable. So I have a few research reports, a couple of diagrams from them that I think are really illustrative of the situation in cannabis in pests, especially in the USA. But there isn't a whole lot of research, so it's actually going to be pretty quick. So this is a figure that has divided the USA into 5 different regions. So you can find where you are on the map if you're in the USA, and if you're not in the USA, then you can maybe consider if these environments are similar to yours, maybe Google it or something. But this first figure talks about fungi, bacteria, and viruses. And they said that about 83% of the responses said that fungi were the first number one pest in all regions. So of all the survey responses. So some of these big contenders are, of course, bud rot pathogens like botrytis, powdery mildew, fusarium, and you mentioned this, septoria, right? And alternaria and cercospora and all of these. There's also oomycetes like pythium and phytophthora, which are infamous pests in many different crops. You can see that fungi make up the majority of pests in this pie graph. As for arthropod pests, so things like insects and mites and that sort of thing, actually, interestingly, 23% of survey responses found that moths were number one of all regions, and it was tied for some others. So we have corn earworm, Helicoverpazea. We have the European corn borer, the fall armyworm, which is really common in many places on Earth, the hemp borer, which is a specialist of hemp, although I think it might go after other plants too, I'm not sure. and the stalk borer. And then about 15 plus 8%, so 23%, they kind of organize it a little bit weird. That's why I'm organizing it a little bit weird here, are the, quote, bugs. So your Hemiptera. Those are things like cannabis aphid, stink bug, lygus, leafhopper, and they also put thrips, kind of like they kind of put them together. And I suppose the thrips are closely related, but they're not exactly the same thing. So Western flower thrips is another big one. This is also something I'm going to rush through, mostly because you do have access to these slides now, and you can pause the video or you can come back to this video later. I want this to be a cool reference for people so they could come back later and be like, oh, what were these pests? Or, oh, let me cross-reference this with the pests I'm dealing with. I really encourage that behavior. So this research is meant for people to use it. But you can see in this figure, if these sections are colored red, then these pests were found in all 5 regions. If they're yellow, they were found in 4. If they're blue, they're found in 3. And if they're green, they were found in 2 regions. And you can also see the percent occurrence from the survey responses. I do want to point out, though, at the bottom right, we have some viruses like beet curlytop virus, hop latent viroid, and other viruses that apparently included in the 30% occurrence range. So that's, I think that's something not to sneeze at. I think this is becoming more and more of an issue. I'm talking with some people who are having lots of issues with outdoor fields or greenhouses with leafhoppers giving them beet curlytop virus. So watch out for that. If you're indoor, you can insulate yourself from these in quite a bit, but not necessarily is it impervious. There's also a research report from Florida in 2024 that went over some major indoor pests in Florida particularly. But I bring it up because Florida is a huge sort of trade area, and it's also a huge inflection point for other species coming into the USA. And several of those end up being pest species. So the major indoor pests that they recorded included hemp russet mite, silverleaf whitefly, impatiens thrips, which is a different kind of thrips than most people usually encounter, two-spot spider mite, greenhouse whitefly, cotton aphid, cannabis aphid, and rice root aphid. So a lot of these I've also encountered in indoor areas as well. And in this figure, we can see that some of these names are colored in red. And that means that they were absent, but they were intercepted. They were intercepted in like trade. They were intercepted in investigations. So one of those is the Old World bollworm, which is a really common and huge global pest, and I think it's going to cause a lot of problems for people in cannabis moving forward if you're outdoor. This is just another example of the different taxonomic groups. So if you're somebody who is familiar with some of these, I've articulated them here on the bottom right. So you've got basically the largest group, the one that I would say people should focus the most on in being aware of, are things like Hemiptera. So your aphids, your scale insects, your whiteflies. But surprisingly, moths were number 2 in indoor Florida pests, which is sort of surprising to me. It doesn't conform to my own experiences or biases. That doesn't mean that's wrong, but it's sort of interesting to see that. You can come back to this graph a little bit later if you like. As a visual, this is what some of these pests look like. And again, you can see in a different way that Hemiptera dominates. And so do other fluid-feeding insects like scale insects, which you can see. Can you see my cursor if I put it here? Or no?
Say that again now.
Can you see my cursor?
Oh, yeah, I can see it.
Okay, cool. So like these are mealybugs, these are aphids here, these are thrips here, more or less. This is like a brown scale. These are beetles, ants. These are also scale insects. This is a leafhopper, lygus, and things like this. Snails even being an issue. Here's an example of what some of those look like. I'm going to move through this pretty quickly because I want these visuals to just be there for people to see. But you can see aphids and scale insects and whitefly for yourself, what some of the various moths look like and their larval immature stages, which is really important for identification. And you can see there's multiple thrips here: Western flower thrips, onion thrips, which are the 2 big ones that I hear a lot in research and also in my own experience, and also chili thrips and echinothrips, which I don't see very often myself, and I don't think they're a huge pest. Here are some of the beetles. I'm surprised to see a weevil here. I don't really encounter a lot of weevils in cannabis, but maybe there's more to come on that front. And just because they're associated with the plant doesn't necessarily mean that they're pests, I believe. So some of these, I mean, the Japanese beetle is a huge pest. The southern corn rootworm is a huge pest, but some species might not be as much of a pest as others. I just want to make that point. Here are some of those other Hemiptera that I was talking about earlier. And then here are some sort of assorted pests that they were finding in Florida. Earwigs, I don't think they're a huge problem myself in my experience. I have a lot of experience, though, with leaf miner flies in the upper right here, these sort of black and yellow ones, especially if you're in a greenhouse where they might be able to get in. They're very small, and they lay eggs inside the leaves, and the larvae tunnel throughout the leaves. So if you ever encountered that, that's what that is. And then we have things like broad mites, spider mites, of course, the false spider mite, and aerophiids, which are russet mites. And even snails were found indoor. These pests are coming from 3 major places: the Neotropics, the Asian Palearctic, and the Indo-Malaya. And the trends seem to show that throughout the decades, basically, we're getting more arachnida, so like mites and things like this, and perhaps spiders as well. Lepidoptera, so moths, are increasing since 2010. But Hymenoptera, or basically ants, are wasps, bees, and ants particularly are going down somewhat, but wasps particularly are going up, whereas the other 2 taxa that belong to that group, the bees and the ants, are lowering. So that would include places like, so just so as a visual, what you might not know is what the Neotropics is, but this is the Neotropics, basically a large section of Central America and basically all of South America. And on the left, I have an example where budworms have been shown to move up into Mexico, for example, for the US, and they have all these resistance traits. And these resistance traits are now becoming a problem for farmers. This is the East Palearctic region. So if you have any plants in your grow space or near where you live that might come from these areas, you could find that out and maybe make an assessment. Well, maybe I don't want those to be around my plants potentially if they could harbor certain pests, especially new ones that are from these regions that might be able to use them as hosts. This is the Indo-Malaya region, for example. So that's parts of India, Indonesia, of course, and southern parts of China, Taiwan, places like this. And I'm going to actually skip through this a little bit because I think it's somewhat ancillary. But these are some other graphical data points. Basically, the kinds of species they found, how many of them, and what percentage of the total they were. So that might be kind of helpful for trend setting. A lot of people have access to ways to analyze data that are far exceeding the capabilities of the past. So having this information, I think, is really useful, especially if you can come to it at your leisure. So this is actually a slide from a European presentation that I'm going to make, which is basically talking about the importance of research. So we're going to perhaps see in the future effective inspection systems, robust quarantine procedures, cross-infection crop risk assessments, and a lot more resistance testing in the future, which is something I'm actually going to be doing very soon, hopefully. I also want to talk about aphids. So there's basically 3 or 4 major pests, and we're going to go over some of their capabilities here. That section right there was sort of an IPM primer. Here's what to think about, especially if you're in the USA. Now, we talk about the aphids. This is a picture on the right of a cannabis aphid with hemp russet mites on its body. I basically have never seen this since here, but it's an interesting point to show that some of these pests can actually mount onto other pests and be more complex than the sum of their parts. On the left, we have an example of a lady beetle feeding on aphids. Many of them are known to feed between 50 to 200 aphids per day, depending on the size and where they are in their life cycle and what species they are. But here's an interesting point. On the right, we have a figure that shows samples for aphid testing, or rather aphid population testing. And so this would be the northwest of the USA, basically, one state. And then we have central North America. And then we have a little bit of the UK, or well, most of the UK, I should say. But at a 30,000 foot look, in a USA context, the bottom line up front is that aphid abundance is actually on the decline, fascinatingly. Mostly these are pest species, which is good for us, but also non-pest species and keystone species, species important for the ecology. You might think, well, aphids are pests. How can they be good for the environment? Well, they were not human pests before, and they do serve a purpose, usually as food for the things that we care about, like biocontrols, or what we would call biocontrols. They're just natural enemies, so to speak. And also, they can be important for feeding things like honeybees and natural bees by taking sap and sugars out of plants and making them available to other organisms, even mammals in some cases. But the central Midwest is seeing the most of this sort of shift in abundance. So lots of this, as I say, these sort of red colors, so minus 50 to minus 31% per year. That's important because there's a, as an example, the woody apple aphid wasp feeds on the woody apple aphid, of course, and when it has resources like access to water and even honeydew, it's 5 times as longevitous. And so its capabilities increase when it has resources or alternative hosts in the case of other parasitoid wasps. So if you've ever used them and they didn't work very well in like a greenhouse or an outdoor setting, or you're trying to attract these parasitoids, there are these sorts of factors to consider. If they're starved, they perform very poorly, as you can see from this diagram. So at a 10,000 foot or 10,000 feet look, we have two sample examples here. So in the Midwest again, an 8.3% per year median drop in aphids. Root aphids like Rhopalcyphum rufia abdominale. Sorry, I usually get that correctly, but the red abdomened Rhopalcyphum, the rice root aphid, is really having trouble, at least in this section, and also other Phorodon species. So Phorodon cannabis is the cannabis aphid, but Phorodon humuli is the damson hop aphid. It's a sister species. It's the most closest related species to cannabis aphids, and it's on a 1 to 30% decrease. So perhaps cannabis aphids are also on a similar decline. It's unclear, although there might be a lot more cannabis in some of these areas, or hop for that matter, so it's unclear. But something to think about as cannabis growers. We might not come across this kind of data typically, but I do, so here you go. Also, if you're dealing with cannabis aphids, or if they're a huge issue for you and you don't have other alternatives, it might be interesting to know that research has come out that CBDVA, or cannabidivarinic acid, if that's how you pronounce it, is a problem for even the specialist cannabis aphid. And we can see that on 7-week plants, as we give these high CBDVA and low CBDVA populations to the cannabis aphid, we can see that the high CBDVA affects the survival of the aphids. That would be these blue sections here, these blue lines, the solid blue line, the dotted blue line, and the dashed blue line. Whereas the low CBDVA genotypes, cannabis populations, the cannabis aphids were much more suitable, or found them much more suitable. Here's another example, adult survival rate plummeting with artificial diets of 0.5 and 1 millimolar of CBDVA. So that kind of shows it that even if you get rid of all these other externalities, the CBDVA is the major toxin here, at least in these examples. Nymph survival was not much better with the high CBDVA, again in the blue coloration here. And also, yeah, so this is also showing a similar sort of function here versus a control diet. As you can see, there on the control, there was really basically no change. Similarly here, but for nymphs. And then we're going to talk about budworm moths. And after that, powdery mildew, and then we'll be able to have our conversation. But I appreciate everyone listening. This is an example of what budworms can do to your flower, especially a very large one that has already developed quite a bit. The moth is in the bottom left, and the egg is in the bottom right of this left picture. And on the right, we can see that the larvae can come in a lot of different colors and sort of shapes. So you got to be careful with the budworm moths in particular. So the corn earworm, Helicoverpa zea, and the cotton bollworm, Helicoverpa armigera. This is what they look like. This is my hand. And then on the right is an example of the moth feeding on some flowers. If they were a Pokemon, I think this is what their card would look like. And I want to mention here and iterate that these moths are like an amazing evolutionary phenomenon. They can move like 2,000 kilometers, which is basically the entirety of the continental USA plus a little, just by themselves. Only 750 kilometers, which is basically half of the continents of North America. But with wind, they can get up to 2,000. They reproduce about 1,000 eggs per female. Their host range includes an excess of 300 species, and they're resistant to things like DDT and Bt bacteria, which are commonly used in cannabis against budworms. So if you're just learning about them now, some of the most common ways to deal with them are already having some issues. Another bottom line upfront assessment is that basically budworms have chemical, Bt bacteria, and cold environment resistance capabilities, or different populations do, that I think are going to be important for us to look at for as cannabis growers going into 2026 and beyond. There's already populations hybridizing and distributing through Central America and the Great Plains of the USA, which is where corn is usually grown. If you've heard of the Corn Belt, they're a huge pest there. So they have defenses against plant defenses and even acaricides, insecticides, and things that normally don't affect them or would have sort of like broader toxic effects. They are resistant to Bt bacteria, like I mentioned earlier, not only from their own genes, but also because they have bacteria that they co-opt from plants, like Enterococcus and Klebsiella, among others, that actually bump up the pH of their gut so high that the Bt proteins that are supposed to kill them neutralize. It's insane. Their pH in their gut is 1 magnitude below bleach, if you can believe it. So that desensitizes and sort of neutralizes a lot of chemical compounds from plants. They can also survive minus 20 Celsius conditions as pupae. Particularly, we find Xinjiang populations in China that can do this, but these are able to move out of China and affect the greater population. And we do a lot of trade with China, and China does a lot of trade with other places. So the movement of these genes are pretty likely. And it's one of the only pests that has the descriptor as megapest in research reports, which is surprising. This cool diagram shows on the right keg pathways of different populations of the budworm. So we have Africa, South America, Europe, South, I believe that is South Asia, South China, Oceania, and Xinjiang is at the bottom. And these are different like genes that have been that are like specific, different types of genes and what they do. That's what the keg pathway shows us. And you can see different populations have different specializations, but overall about sort of a cacodemoniac 18,455 genes related to adaptation were found in this research. Two major sections: toxin processing, which I can't stop talking about, and plant sugar processing, which gave them their cold resistance ability. This is like from the GIF image from before. This shows some of the developments of these resistances, and if you are in the USA, you can plot where you are on the map and where some of these resistances may come from or are already existing. So we got a few examples of Bt resistance genes found in 2019. Pyrethroid resistance genes. In 2016, we found more Bt resistance genes, and I bring up here that there's a 20-year timeline between the use of Bt crops, if you're familiar with genetically modified crops with Bt proteins in them, that is what those are. And it only took 20 years, less than my lifespan, for these moths to come out and become highly resistant to those very proteins that are used in GMO crops. So that wasn't very effective. In fact, it was the opposite of effective. We have other pyrethroid resistance genes coming out of places like Brazil. And in China, we have a lot of these Bt, cold resistance, and toxin resistance genes, and even the microbes therein. This is a example of the Corn Belt. If you live in this area, then you probably have heard of budworms. If you haven't, though, they're a huge issue. And I bring this up because I think some of these traits may concentrate in this zone, and then sort of expand out to the rest of the USA. So even if you don't live in this area, I think it's important to be aware that you might be affected by this area. And so maybe be aware of what you can do to maybe stymie that or mitigate it, even at a regulatory level. Some of these magical microbes that help budworms to sort of drill in my point. So these are very adaptive organisms we're dealing with, not just Enterococcus and Enterobacter, but many others like Commensalibacter and Acinetobacter. Rhizobialis was even found to be um, correlated with the gut of the budworm, which is crazy because most people know those as sort of like soil plant microbes. Just to give you an example of these microbes' capabilities, other Enterobacter, like Enterobacter horimeici and Enterococcus cassiflabis, are associated with multi-drug resistance in hospitals. So that's not good. And so is Ralstonia and Acinetobacter as well. There are other species here that I've noted that are known for like degrading toxic substances or even digesting things that are normally indigestible, like the aptly named Cellulomonas that degrades cellulose, right? And then here's an interesting research report that came out in right this year, 2026, a few days ago. I've put it in the presentation for us. They compared domesticated versus wild cabbage, and they found that in wild cabbage, dirtledore, that this cabbage butterfly larva, Mamestra brassicae, was when it fed on the plant microbiome reacted and increased the plant's defense against the caterpillar. It took about 2 weeks for this process to occur, but once it did, the caterpillar's weight went down, which is what we use as a sort of indirect way to tell that the caterpillar either stopped eating, wasn't able to digest the food very well, or was inhibited by feeding from other pathway effects. However, in the cultivated population, Rivera, the opposite happened. The microbiome reaction had a negative effect. It actually increased susceptibility, and by doing that, it decreased the jasmonic acid pathway, the JA pathway, that the plant uses to react to caterpillars. So we might be aware, and I hear people talk about all the time how important the microbiome is, but it has to go further than that. We have to be aware of what those microbes are. There's technologies that allow us to understand this a lot better. But this we might learn that plants can have a plant response or defense response, but we have to be aware that our domesticated plants may not have those capabilities. So we can't just assume that plants are operating on all cylinders, especially after years and years of human intervention. So this was an excerpt from the paper. I'm not going to go into it all the way, just enough to say that caterpillars, like budworms, they can shape and benefit from the plant microbiome. But the defense response, even in the wild population, was slow. It was like 2 weeks. A lot of cultivation scenarios require a little bit more zest in their response. And also, pest density inhibits resistance. So, as they added more caterpillars, then the defense response by itself was not enough. And so caterpillars en masse can, and other pests for that matter, this is the point of the everswarm, they can have a multiplicative effect. Even when one individual is already pretty capable, 20 of them are even worse. So we can't sort of presume that wild type examples are going to be the same as our cultivated, and even between cultivation, it can be very different. But what's interesting is that they tried this in different soils, and they found that the effect was soil agnostic. So it was really certainly the microbes and not necessarily the soil. Interesting point. And these are examples of the caterpillar weight going up on the domesticated population of the plants. And then we have examples here of, on the right, caterpillar weight going up in the Rivera and down in the Dirtledore. So what this sort of light green, sort of lime green coloration shows. So the jasmonic acid pathway went down within about 3 hours, and in the case of the cultivated population. So that's a really quick reaction for the soil microbiome to negatively affect the plant. way quicker. So within 1 day of getting the pest, you're already seeing problems. The last one we're talking about is powdery mildew. In cannabis, we mostly deal with Golovinomyces and some other powdery mildew species we'll get into. But if you've never seen powdery mildew, this is an up close and personal picture of the hyphae and the many fruiting bodies that it produces. Here's another example from a recent research report. You can see the hyphae growing all over the trichomes. They're unabated. There's a question about whether powdery mildew is a systemic pathogen or not, but it's not. It only exists on the surface level of the plant tissue, and it only penetrates into the plant cells that produce sugars from photosynthesis, and no further. An important aspect of powdery mildew that I think people never hear about, which I think is pretty awesome, is that they have a close relationship with another group called the Arachnopezzaceae, or the spider puff family, maybe if you translate it. I like spider puff. Basically, they have a lot of similarities to what we might consider beneficial soil fungi. So they have shared genes that break down cellulose in plants. So normally these other fungi, they would have used that to break down the nutrients and then make them available to the plant. Powdery mildew shares those genes. They also have a shared way of interacting with the plant through certain chemical or other hormonal pathways. The MLO genes are an example of this, and I've spelled response wrong somehow. Rare slide L. But plant MLO genes are important to know because basically these are susceptibility genes. So you might think you give plants nutrients or other sorts of benefits, and it means that the plant can never be negatively affected. But in fact, pests often do exploit these pathways that they have. For example, normally the plant would use them to interact with mycorrhizae or other beneficial microbes. But if the MLO genes are not there, then both powdery mildew and those microbes cannot connect with and communicate with the plant. So it's sort of a double-edged sword if we are able to get rid of MLO genes, and in some populations they have. For about 100 million years, powdery mildews have had a temperate distribution. We can see here this research report shows the many examples of species that we know, and wouldn't you know it, Eurasia has a lot of it, Asia has a ton of it. Some people would consider the cannabis origin to be at or around the sort of area in Eastern Eurasia, and probably moved out quite a bit after that. And also North America, and the USA in particular, has quite a bit of species. So, not great, but it's important to be aware of this. If you're wondering why you're having so much powdery mildew issues, that could be part of the issue. And cannabis probably developed with powdery mildew for about 47 million years, since its origins. Maybe a little bit more, plus minus a couple million years or so, but that's quite a lot of co-adaptation. There are other powdery mildews and other Cannabaceae, the family of cannabis. So the hackberries or Celtis. There's two species like Erysifi cousinoi, which colonizes Celtis australis, and Pleidocata shiriana, which colonizes Celtis blondii. And there's also pigeonwood or trema that also has a powdery mildew as well. So those might also be able to colonize cannabis. We've not looked into it. Why are powdery mildews able to do this? Well, you never fight the same spore twice. They mutate all of the time. They have these sections called dynamic genomic compartments, basically sections of their genome, the collection of genes in their body that allow them to colonize plants, these virulence genes, and they're mutating all the time. Every spore gets a little bit of a different mutation, and if they're really good at what they do, they go on to make millions more, and if they're not, they die. And so this selection pressure has honed powdery mildew to be really, really good at getting in there and getting that first bit of nutrients and to manipulate the plant. The three main species we want to look out for are here in sort of a light green color. Golovinomyces siceracerum, which is lettuce powdery mildew. It has over 100 host species, and it's also really prone to hybridization with its own species or other closely related species. Golovinomyces ambrosiae, which is the immortal quote-unquote powdery mildew if you translate the species name. It has about 25 host species recorded, but it's the understanding of what populations are or are not this species is a little bit fuzzy. And then we have Podosphaera macularis, which is hop powdery mildew. We've also found it on strawberry and caneberry. So these are three species, and the plants that these species colonize might be something that a entrepreneuring cannabis grower might consider in their backyard or on their property, however you're growing. And just so you know, I'm not kidding around here. The Golovinomyces siceracerum genome, as you can see, I've highlighted here in green. About 60% of its genome are these virulence genes. Almost, more than half of its existence at a genetic level is meant to manipulate the plant. So it's pretty good at what it does, and it can't do very much more than that. I like this figure a lot because it visualizes what I was just sort of talking about. So if it was a little bit complicated, basically I've highlighted with these green lines different groups of species, which are known as genera or a genus, like Golovinomyces or Pliocata or Podosphaera or Erysiphe at the top here. These are all genera that could have species that might be able to colonize cannabis. And you'll note that, like I mentioned earlier, that Golovinomyces has a blistering 335 number of host genera and the species of those. And then of the type of host they colonize, they tend to be the purple here, this herb type. Cannabis is an herb, right? But it's a woody plant too, and certainly they colonize tree and shrubby plants as well. But also Podosphera here, 198 host genera. So these are all plants, and also able to colonize a lot of different sort of herbal or herbaceous type plants. This is an example of melon powdery mildew at the top and melon cultivars at the bottom. And, I bring this up because every time we've made a cultivar that's resistant to some of these populations at the top, a more capable strain of the powdery mildew PM develops. And then we have this sort of spear and shield problem. We've had it for almost 100 years here, as you can see. So we have to be cognizant that even if we get resistant plants in the future or recently, you might have heard research of resistant cannabis populations out there. Well, how robust is that resistance going to be? It's unclear, but you might take examples from other crops and know that even really capable resistances, now a lot of these were a long time ago, so I don't want to totally equivocate this, but you should just be aware that, resistances do break sometimes. Powdery mildew, like I said, does not colonize further than the sort of upper layer of the plant's tissues. There are three basic ways that the cell of a plant defends against powdery mildew. So if you're aware of this, you might be able to, as a grower, do things or use things that allow you to maybe out manipulate the powdery mildew. For example, when the powdery mildew spore latches onto the top of the plant, you can see in the upper right here, there's examples of non-host resistance or incompatible interaction. That's when the powdery mildew develops its little hyphae, tries to go into the plant, and the plant does not allow it because maybe it's able to sense it outside and render the right defense response, or maybe because the powdery mildew, in other cases, was able to get further in, create this development called a hostorium, basically a sac where nutrients are supposed to be absorbed and manipulative molecules and factors are deployed. So what plants do is if they have a competent response, they will produce these structures that will choke out the hostorium. Basically, they glob on to the sac and then they basically suffocate it. And that's important because that sac is where the nutrients are going to help the powdery mildew develop. But if they're able to successfully do this before the powdery mildew goes to the next plant, and if they're able to prime other cells nearby, then the powdery mildew colony either doesn't develop very much at all, doesn't develop entirely, or is challenged along the way. all these things are good things, but not all of these things are the same thing. You're still going to have maybe some powdery mildew, and so you might need alternative options. But it's always good to have a resistant plant versus one that is not. What's really cool is that this guy, Ralph Huckelhoven, found that in barley, powdery mildew infection caused them to change their aromatic profile, and even answered a question that I had on X about methyl salicylate, I believe. Yeah, methyl salicylate being important for priming the salicylic acid pathway, right? And to disrupt powdery mildew. So that's really cool. Reach out to these scientists, reach out to these researchers. They love talking about their research. You can get a lot of cool insight and information. But these light blue sections like heptane 2,3-dimethyl, boranyl acetate, heptane, these were upregulated quite a bit. And then the really crazy part, they flowed through the canopy and they primed other barley plants. So then when the powdery mildew came to colonize them, they were already primed, and the colonization was not nearly as effective. Pretty cool. Something that maybe cannabis growers can utilize as well when we get the right information about how that works. Another example of resistance being kind of a spectrum is that this report looked at hemp populations getting colonized by powdery mildew. And we can see, on the left, we have really susceptible populations relatively. And then on the right, we have much less susceptible populations. At the top here, in row 1, that they were, if you see the circle, they were colonized by the powdery mildew. And the size of the circle shows the severity of that. If they were in the section 0, then they had less than 0.5% of their sort of colonization quotient. So that might mean that those populations did a lot better, but they still got some colonization.
Oh, hey
That was
Matt, can
in
I ask you a quick question? Sorry about that.
Absolutely. No, please do.
On the last slide, or showing the set, or I'm sorry, one
Oh,
skip
this
one
one.
head. There you go. I noticed the ones that are less susceptible tend to be like Futura and Fiber. Those are hemp fiber strains.
That's right. That's right. Fibror Futura 75. I was talking to somebody about this recently. Did you have some insight?
Do you think it's like the lignin content, perhaps, or something of that? So I mean, there's not as much vegetation, but I was just wondering. And plus, like
It's
those like cultivars are like they've been used for ever, you know. So I was just like wondering, I'm not sure. I just know there's like less vegetation and more lignin, but I mean, we grew 70 acres of this Chinese variety Yuma last year. No pesticides or anything, and like we did not have like any sort of infection or no pests, nothing. So when you show that graph, I was just like, okay. So
That's so cool. That's exactly the kind of insight that I want to hear when people hear this presentation. That's so exciting to hear, honestly. That's really cool. I like that because I was just talking to somebody about this and I was trying to understand, and I think it could be a lot. To answer your question, I think it's possible. Lignin, it's so funny you say that because there's a section where I think I took it out. Yeah, I took it out. I'm just giving you a little preview. But yeah, where I talk about exactly this, about resistance for powdery mildew and lignin being part of that. Very, very possibly. It could also be because, powdery mildews typically don't colonize the stem tissue, but it could be that there's still higher lignin content or cellulose content, or maybe even that they even bioaccumulate metals and other compounds more, and maybe they put it into those ancillary tissues more than the other plants do. I think that's a really perspicacious suggestion. You can see here that Fibror 79 also is pretty low. Auto 2, very low, ACDC, Carminola, cherry wine, which I've grown quite a bit, and photo CBD is closer to the susceptibility range. I just want to say also, when people say that their plants are resistant to pests, you should ask them which pests. If they say all pests, you should run away, because that's not really how resistance works, and I hope that I've made that case here. But these researchers doing real resistance work in crops where lots of time and effort are on the line. They use these methodologies like area under the disease progress curve, or AUDPC, as you can see here. Those are the kinds of quantifications that really allow us to dig deep and kind of understand a relative spectrum of resistance to one pest, let alone many others, and environmental stressors as well. For microbes, for example, this research report looked at what's now called Peribacillus. It was bacillus, but Peribacillus frigotolerans C1141. This particular sample had thousands of growth promoting genes. I've articulated here 22 direct affected gene types, doing things like solubilizing phosphate and potassium, degrading xenobiotics. So those are like compounds that are alien to the plant. So some of them could be toxins, for example, and also the production of various metabolites. Then there were genes like related to abiotic stress neutralization, plant-derived substrate usage. I'm not sure what they actually mean by substrate here. I keep saying it every time on the slide, hoping that they just mean subtract, because I've heard people use that word as well. So if anyone has insight on that, I'd love it. But it seems like they're using extracts perhaps from the compound. So I'm not sure if it's a typo or if they're trying to say something else. I couldn't find it in the paper, but they sure have a lot of it. Over 550 of these genes. They also had a lot of genes related to induced systemic response and systemic acquired resistance, which was what we were talking about just now. And then also things related to things important for the bacteria, bacterial fitness in general, ecological capability, spore production of the bacteria. This is the kind of quantified microbiome research I think that growers should demand and sort of look forward to, because some individual microbes are incredibly versatile, and others might do the same job better. You just don't know. Here's an example here. Serratia plymoutheca is another sort of growth related microbe that people like to use. And you can see that based on versus control on the very left here, Serratia was really good, but Peribacillus blew it out of the park with biomass increases of over 260%. Huge, huge. So you can see that stalk diameter increased by 96% and the height increased by 43.3%, controlling for other variables. So that's a huge change. Other microbes were found to affect different traits. Different bacilli were found to affect things like germination rate and the development of true leaves and secondary roots. Some of them also had negative effects on some of these traits as well. This was a collage of various bacteria that were considered sort of correlated with the cannabis microbiome, going through a bunch of accessions. This was a lot of work, and basically, I don't bring it up because I expect people to learn all the different names, but I bring it up because I want to say that 38 phyla of bacteria is a large amount, and 800 plus genera, of which underneath them there's probably several species each on average, is a pretty insane amount to sort of collect. Some of the major ones, though, are going to be in the Bacilliaceae, so the Bacillus genus in particular. Pantoia was found really correlated. All of these that have like a light green to them, they were sort of more represented. And those that are closer to brown or green, or like sort of this muck green, are closer to zero, right? Pseudomonas and Ralstonia were also sort of household names that I think are very famous. Ralstonia, many pathogens are associated with Ralstonia, but there are also a lot of really beneficial to the plant species as well. So there's sort of both of those. Pseudomonas is also very famous for having both really good and really bad bacteria for plants, so to speak.
So if you go back to the last slide, were
Yeah,
these like just the microbiome from the seeds alone, or?
yeah, so this was the seed microbiome, the spermome, or the spermosphere, if you will.
Oh, okay.
Yeah, so this research was specifically from the seeds. I'm glad that you brought that up because I totally glossed over it because I've given this slide a bunch of times. But yes, that's actually right. So this research, you can check it out. I think I have it cited somewhere. And if not, if you look up seed microbiome research for cannabis, you'll probably come across it.
Now, are any of those systemic or inherited from like the actual plant, or is it just kind of like what's there in this, like the
Some of them
CDO
were
license? Is
vertically
there anything like
transmitted.
systemic by any chance?
Absolutely. Some of them were definitely vertically transmitted. If you can believe it, I cut down slides to make this a lot quicker, but there was
Oh
a
no,
slide
I
that,
totally believe. Because I used to do metagenomics.
there you
So
go. You get it.
yeah,
You get it. But I have a slide, and the report goes into it too. So I really encourage people to take a look at it, especially because the pictures are nice. Even if you maybe aren't, big up in the terminologies, the diagrams are very illustrative. Um, so the core cannabis microbiota across the different domestication grades that they were looking at were Bacillus, Pelomonas, Ralstonia, Staphylococcus, and Pseudomonas. I think like 4 out of 5 of these are really, really famous genera that even people who are not into metagenomics or any of this stuff have probably heard of, either in microbial products or in various research reports. So that's kind of interesting. This is a report. This is a diagram from a cucumber paper, and I make this point, about equivocating biodiversity. And I'm going to close out with this point and some considerations, which is basically that we have to consider, excuse me, what is beneficial and what is a beneficial consortium? Is it one that's biodiverse? Maybe. Here's an example of a native rhizosphere, a complex network. They're trying to represent 3D space in 2D. So that's what the lines are supposed to show, is like a connection. But larger nodes, all these circles are nodes, means there's more community interaction between these different groups of bacteria. Smaller nodes, less community interaction. And the lines show 3D connection. So, I mean, is it biodiverse? I'd say, yeah. Proteobacteria, actinobacteria, tons of them. Is it plant pathogenic? According to the paper, no. Here's a dysbiotic rhizosphere. It's reduced and somewhat transformed. Is it biodiverse? Yeah, I would say so. It's about minus 27% of the total nodes, but it's about 5 times higher community connectivity. So less microbial groups doing more, or I should say, less microbes in those groups doing more connection. And also, some of these groups are not as present. We're missing, if you go to the before slide, acetobacteria, and was it chloroflexa? Yeah, chloroflexata. So like, even if you have lots of different microbes, it might not be good for your environment. And as people get the ability, and maybe you can speak on this, if people get the ability to actually look at the genes of their plants, make their own informed decision, look at the microbes in their soil, on their plants around their area, they're going to be able to make informed decisions. They're going to be able to see that you can put thousands of microbes and it won't matter, necessarily.
oh
Here,
yeah.
this dysbiotic group, we have a higher, 2 categories that were, not represented in the other one. One of them is even, oftentimes they include predatory bacteria, so not necessarily pathogens, right? You were going to say something.
Oh no, I was just agreeing.
Oh yeah, yeah. No, I appreciate it. Yeah, but here in this example, earlier we had like 31% of the group being proteobacteria, and now we have almost 50%. So more is not necessarily better of the largest amount of those microbes, right? Here, and I don't know if I don't know if the amazing, like, sort of like a symmetricality of this diagram is just an artifact of how they decided to shape the picture or not, but it looks a lot more prettier than this and this. But I don't want to let the diagram speak for itself. This was a native rhizosphere of cucumber without any microbial inoculation. Is it biodiverse? I would say yes. Is it plant pathogenic? According to the paper, no. Minus 9% total nodes, similar community connectivity, similar composition. Also, even had some of those guys from the other one that was dysbiotic and no problems. So I bring all this point to say that you got to know your pests and you got to know their vulnerabilities. You have to have a robust IPM. You have to have proactive and reactive measures and options. You want to lose the land. You want to use the landscape to your advantage. And even if you're looking at research like this, or you're looking at presentations on those research, even if I'm an okay guy and I'm interested in showing you this information and I'm curious for people to like learn more, review and analyze the educational content that you consume. Trust but verify all sources. All information is equivocal. Platitudes and anecdote do not equal data. And even if I've said cool things that you might agree with, you should definitely check to see if I'm right or if new research has come out. Don't just believe in ideologues, right? As George Washington would say when he was a major in the British Army, there is nothing more necessary than good intelligence to frustrate a designing enemy, a thinking enemy, and nothing that requires greater pains to obtain. Is our pests a designing enemy that thinks? Well, genetically, I think they are. I think that you could make an argument that they hold a sort of memory, so to speak. And certainly there's behavioral reactions as well. But yeah, my point here is that the Everswarm is constantly evolving as Sun Tzu says, or Cao Cao, or some of these other people who, if you're like me, you've studied military science and history, you know that the art of war has been written and rewritten and reviewed quite a bit since it was first written. So who knows who really said these words, but if it's Sun Tzu, he said victorious warriors win first and then go to war, while defeated warriors go to war first and then seek to win. Win in the temple first with your theory and plans and practice, and then win in the field. It doesn't mean that everything is planning and that nothing is execution. A lot of cool quotes about that too. But I think it's really important. So, non cognosis si non id viris, non lucebis si non nuceris. Or as Smash Mouth would say, you'll never know if you don't go, you'll never shine if you don't glow. Research is paramount for better options. Support your researchers, learn research, be aware of your pests. Lots of cards in play. Fatal in combination, severe in isolation. Lots of combos, lots of complex names, but lots of robust capabilities. Have respect for the pest. And that's the presentation.
Awesome. That was awesome.
I hope so. I'm glad you liked it.
Let's see. There we go. Oh man, I got so many questions, and
That's good.
and
I'm
our
glad.
viewers have some questions too.
Good.
Just I'll go ahead and ask one of mine off the bat. But the hemp borer, is that throughout the USA? Is it still kind of out west, or?
I wish I could give you a better answer to that. I think it's not isolated to the West, but that's about as close as I could give you to good information.
So I'm out here in the Midwest, man. It just seems like new stuff's popping up all the time. Like corn borer, it's always been here. It seems like a thing that caught me about the aphids populations dropping. Why is that? Is that just because of climate, pesticides, or?
It seems like their life. I didn't go into this point, but it seems like aphids have a very complex life cycle, and most of them are specialists. So they only feed on one species, or they've speciated onto a specific plant species from a more generalist, progenitor. But basically, in some of these, like rice root aphid, okay, the natural environment that exists in East Asia, it feeds on herbaceous plants in the spring and summer, and then shifts to reproduce on like prunus, which are like apricots and cherries and nectarines and things like this, and some other tree-like species. So those are two totally different plants, two totally different areas. If one of those goes away, then the population has a truncating effect, and so that seems to be the problem. They're having such a complex life cycle that's reliant on their plants being there, that when they go away or something happens, that's what happens.
Okay, see, then
And the temperature might have an effect too. The temperature may have an effect as well. There's a graph that I took out of this slide section that actually showed that I think temperature or weather might have been the highest, but also next to it was reverse correlated. So maybe that's helping them out more often. But yeah, good question.
It's scary because not all aphids are bad, so it's just like,
right.
Hey.
Yeah. I'm glad you make that point and reiterate it, because it's true. Like, aphids, there's a honey that I got recently from it's made from pine trees. But what's funny about that, James?
Pine trees.
Yeah.
Oh, I
What's
know
weird
that
about making honey from pine trees?
making honey from pine trees because you need bees, right? Don't
What
you?
else do we need?
Uh,
You need flowers,
flowers.
which pine
Yes,
trees don't
yes,
make.
yes, yes. Of course.
Yeah, sorry.
Of
Sorry.
course.
But so where does the sugar come from? I've answered it. It comes from aphids. Yeah, yeah.
Yeah,
And then
because they
these
secrete.
honeybees,
Yeah,
these
yeah.
natural and other native bees, they come up, they drink the sugars. They're like, wow, I would be dead without this in the wintertime. So, aphids do, that's just an example, and that pine honey is made from bees who have taken honeydew from aphids. And it's really nice, honestly. A lot of, it's very terpinolous and piney. I really enjoyed it. But, that's just like one example of many.
And isn't there like some fungi that help like the aphids like make like honeydew and stuff as well? Is there
I
something
think
along that lines?
Well, they have they have microbes in them, usually bacteria. There are some yeast symbionts in some species. I have an aphid analysis video that goes into that at length for those who are interested in how those symbioses develop and what they can do. But a lot of bacteria help them produce essential amino acids,
Hmm.
and they produce, or they also give them vitamins, and I think they also metabolize some other things. So yeah, aphids would not be able to feed on just sugar sap and get mostly sugars without those microbes for sure.
Speaking of aphids, do you know anything about this? That's kind of in my neck of the woods. So I was like,
The University of Kansas professor that studies aphids thinks Kansas aphids were bioengineered good times. I don't know. Maybe they made a joke. I don't know. Well, I mean, what's your definition of bioengineering? If you mean like with genetic modification, I don't know if that's supported.
CRISPR aphids, right?
Well, in my slide where in my master IPM about aphids section that I constructed recently, um, I have a section that talks about how DARPA had an Insect Allies program in 2016, which I will ominously state has completed, whatever that means, which was looking into using aphids to inject plants with microbes or maybe viruses that were able to actually not to weaponize them, or I guess defensionize them. Really, it was actually as like a way in case there was like a huge virus or some kind of pathogen that would sort of endanger the food supply, the security of the U.S. So the idea was that these aphids might be able to be used as like a kind of a living syringe, right? But there's all kinds of problems with doing that, and I think that I don't want to speak for the U.S. government, but I imagine that they found that to be pretty difficult, and they probably have better ways to do that kind of a thing now.
So they're worried that they're gonna breed with like the wild type population and cause some issues there. Or
Or what if the like defense organism that they use does not operate as you think it's going to, like
I'll go ahead and ask this one here in a little
oh yeah.
bit.
Oh,
Um,
okay.
actually, go ahead and show this one.
I've never seen PM on cannabis. Why is the cannabis you just did today? Why is the cannabis unaffected by comfrey powdery mildew? Powdery mildew finds its own plant species to munch on. Yeah, I mean, like, so, like, that's a really salient point, actually. You might see powdery mildew on the street and think, wow, I better wash my clothes. Now, I really actually applaud this sort of thought process, but it's possible the powdery mildew isn't going to affect you. And it might be right next to your plants because it has that non host resistance. That powdery mildew just doesn't have the genes and proteins that are made for manipulating that particular host physiology. But there's examples in other research where, um, if you turn off certain genes in the plant, or if you neutralize certain defenses in a plant, other under experimental conditions, other powdery mildews can still infect the plant because you've gotten rid of one of those barriers. So really, it's a very tiered, tessellated system.
I was because I mean they kind of go like, so I mean, you can even have like two separate cultivars. Like one's really susceptible PM, the other one's just fine. They could literally be like right next to each other. So I was wondering if that goes to
Absolutely.
that point, or just even like resistant genes within the particular cultivar or what have you.
I mean, the variability of it, I think, I mean, one of the points I wanted to make was that, like, it could be environment. It could be the powdery mildew might be hypovirulent in this case, in this example, I don't think that's the case, but time, maturity of the plant has a factor, the priming of other plants. So if you grow, if I don't know how many people in their experimental protocols are like isolating each plant so that they don't prime each other when you colonize. You know what I mean? So even in those examples, in a field, one plant might get one susceptible individual might get the powdery mildew, let's say, and then it sends off all these volatiles, and then the other plants, and maybe some of them are more resistant anyways, but some of them might not be, and maybe they are primed, and then they have less of a problem. But you can't visualize that with the Mark 1 eyeball.
True, true. Good insight, man. Uh, Budworms, fire.
Point blank annihilation. I
Oh, yeah.
agree.
Man, those things just want to survive. Like,
Yeah, I mean, as you can see, and I made a big video on budworms too, so you guys, if anyone's interested in that, you can check it out. Um, yeah, it's like it's a very interesting pest. It's a very interesting organism in general.
all right, and like the new hot thing, a lot of people thought it was Septoria, but it's actually Alternaria.
Well, there's all kinds. There's bipolaris, Cercospora, Septoria, Alternaria. Um,
Because like I've
all
known an Alternaria or Naria, but like there really hasn't been like a too many documented cases of it infecting cannabis plants. And they're like Brandon Russ noted something's working with like medicinal genomics right now. But some of these are just kind of hard to tell apart unless like take it to the lab or sequence. And
I do
Alternaria, is
know.
that host specific or is it generalist or?
It's super generalist, super duper. That's part of the problem. It also has a very interesting like life cycle that's different than like powdery mildew or fusarium, somewhat, especially powdery mildew, which is that it can live on living tissue, it can live on dead tissue, and it can subsist in the soil for a long time. And so the problem is that I get people who were growing fine greenhouse, outdoor, whatever, their region was all right, and then suddenly it sweeps in one year, and maybe it was a small problem, but they were able to mitigate it or cut it out or whatever. But then once the substrate gets colonized, then you have this big issue because they're still there. They never went away really, and they've got thousands of spores in your area. I actually happen to have for you, since I am just such a people pleaser. I happen to have this. This is an example of some of those species, some of those genera: Septoria, Bipolaris, Cercospora, Anthracnose, Curvularia. These are all cannabis. These are all leaf spot fungi, and possible likely cannabis leaf spot fungi because they're very similar. They have similar capabilities. Um, there's some research about them, and in Kentucky, this might be what was being referred to before. Maybe that was cannabis aphid, but yeah, they are interesting. This is actually going to be partly for a Missouri event, the Common Ground Fair that I'm going to be going to. So, yeah, you guys can check this out. Check it out. Even we've known about Septoria on cannabis for a long time.
Oh yeah.
Yes, a long, long time. This is Taima Shiro Boshibyo, cannabis white star disease. Yeah, anyways, you guys can check that out. I'm not going to get into all of that, but yeah, there's a lot of information about it, a lot of research. But
Another
unfortunately, there's not a lot of solutions. So they're looking at it, but like I wish I could stand up here and said they came up with something, but all the like biological or like cannabis friendly things that they tried were not very good, and even the fungicide, like the noxious stuff you shouldn't be using, was not particularly effective. So that's scary.
And do you think just see like we're identifying certain genes and we're just gonna CRISPR the hell out of it just so like it'll survive, or where what are
I
some
think
things you see us
I
doing to help
I'm
combat it?
probably not. Oh, say the last part. I talked over you.
Just like what can we do to perhaps stay ahead of it? So I mean, I know
Right.
it's best to be proactive, but
Well, I
it's a game of
think
cat
that
and mouse. Just it's
yeah.
evolution.
My problem is that I think the issue is that strategically there's a few different. This is actually a core tenet of the Everstorm question, right? Like, what are the options? Now you've told me that pests are this terrible problem. So what's the solution, Matt? What's the solution? And the solution is we have to have lots of options. The solution is infrastructural support, which we're not getting. The solution is that us as a community is aware of what these issues are, because in some places people have been able to get away with, and I don't mean this in a bad way, it's a good thing. Getting away with this is a great thing, not having certain things in place. But now, things have changed. What maybe greenhouse is not viable anymore because of your region, or maybe it is never really viable and you've just been lucky for a couple of years if you're just starting. People don't even have like a pest history log or ID. There's no data for people to make informed decisions on. But yeah, I think that the if I was like in charge of it all, I would say we've got to do this sort of research, and that's the kind of R&D work I'm going to be doing in Europe hopefully soon. Not for Septoria necessarily, but pest resistance testing and quantification. If you've ever had someone be like, my plants are pest resistant and lost a bunch of money, give me a call, because that is the impetus for this particular problem.
Nothing's resistant to everything.
Nothing's resistant to everything. There's no free lunch in physics. Something is happening. Many things are happening together um, that is disallowing or allowing the pests to get in there. So you got to find out what that is. And I'm excited, man. It sounds like you have a background in some of this stuff.
Yeah.
Do you share my opinion that, like the individual, there will be like a great democratization of like gene analysis capabilities?
Hopefully.
It's okay to disagree. If you don't think that's going to happen anytime real soon, you might be more informed than me, honestly.
I think it'll happen, but like latter.
What
But
makes you say that?
what's that? And when you mean democratization, do you just mean like giving away
Oh,
the
I'm just being
information
fancy. I just
freely?
mean, I mean that. Sorry, yeah, it's maybe a touchy word to use now, but what I mean to say is that everyone has one of these, but 30 years ago it was Star Trek. It was science fiction or whatever. So in the same sort of way, I wonder if people will get maybe not the exact same as like a spectrometer in their living room or something, but I think people will have access to like really cool technologies that will maybe allow them to be like, hey, you said there was a powdery mildew resistance gene 1 in here, and I didn't find anything, man. So
Can we
what's up
do
with
like
that?
sequencing out in the field, like some nanopore sequencing? I think. It's like,
Yeah, like that's a great example, right? There's nanopore, and I've heard some other ones like micro something or other, microboard or whatever. But yeah, exactly. That's what I'm trying to say. But maybe something. I know that those have strong limitations.
oh yeah.
So I just mean like, but something like that, I think will happen, and I think people won't be able. People will argue also about whether somebody's cuts or somebody else's, and all I'm saying is that somebody
Oh,
could at least say
sequencing.
somebody could sequence it. Yeah, that's what I'm saying. People could just be like, or maybe they're like, you took. Could you imagine? I'm just predicting in the future now. Someone's like, I bred these microbes in a vat in my own fermenter, and they're specific to me, and I grew these and I sell them out, and you have my microbes or whatever. Like,
Okay.
I can see
Yeah.
people.
I mean, that's never gonna go away, but I think like open source. I mean, because like I mean, we have like GenBank and Blast and like all these,
Yes.
but not everything's. But even stuff that's like loaded up to it isn't exactly like it's free to like look at, but you're still gonna have like your microbe or your plant that patent. Um,
Yeah.
I just I see sequencing just to kind of settle disputes. and
Yeah, that's what
blockchain.
I'm
I
saying.
mean, like medicinal genomics kind of does it already.
Right? Like, it's already a thing, right? As a
Yeah.
service, so
I mean,
yeah, I
hopefully
think.
everybody's cool sharing information and not trying to screw each other over and monetize it, you know? Because like
Well,
you can
my
see,
hope.
like find something like a certain sequence, right? Oh, hey, I'm gonna CRISPR it into like this microbe or this plant and use it as like part of my cultivar or whatever microbe I want to like release in a product. I can kind of see that kind of happening with some bad actors,
Yeah, I've heard
but
I've heard some talk about this. Yeah.
but I mean, there's always gonna be bad actors, but the future, I see it. I see good things down the road, but there will be some bad actors. So I mean, like
Yeah.
we're not. We need to look at something because, like, as you're showing, like throughout your presentation, like, oh, hey, there's all these like groups of insects, like with different like resistant traits, and they're all just especially like the moths. Perhaps they're all going to combine and they're going to share all these different resistant traits, and you just have like one super bug.
Yeah,
Would you
that's
say
kind
like
of my
something like
point.
that? That's what I kind of
Yeah,
see.
yeah. I don't want to be the bearer of bad news. But yeah, and I think your point about selling disputes, like my hope is that it would be like maybe it would be untenable to lie. Because like it's like there's no reason to. Someone could just spend, whether it's a service that they pay for or they do it themselves or they got a buddy. What I mean is that like people will be able to find out in a way that you weren't able to find out previously. And so if you lie about something, or if you say that you have proof of something because everyone expects it, because that's the thing. I want the standards to go up. I want the grower individually to be like, prove it, prove that your marketing is true. Why should I believe you? For any reason, do you have I hear about like microbial products that don't have the microbes they talk about, or they're dead on arrival, or so I just believe
It's funny you
I just
said that because all right,
yeah,
that's a
why?
different research project. That's a different.
Okay.
I can
Okay.
tell you behind the scenes, but
Well, that's great. That's great. I'm glad. I want people to work on this stuff. So that's excellent to hear.
not gonna throw shade on anybody, but just like, yeah, you're right. Because like a lot of stuff on the shelf arrives to you, and it's DOA kind of.
Yeah.
So I mean, if
Or
it's
in
sits on
some
the
way
shelf, like
inhibited.
those microbes are. I mean, you can lyophilize it, powdered form, but even then, you still gotta wait for those like communities to, like activate and stuff. But
Yeah.
which I don't know, when it comes to microbial products, it's a little bit touchy. But
Yeah, and like I'm not, I might be saying things that sound right to me based on what I know, but it might
then you'll
not
have some
be.
that's throwing like a lot of bacillus species, like when they say like it works for this, like but when you look at the
Yeah.
literature on the organism, it's not really meant
Right.
to. Stuff like
Yeah,
that. There's a
exactly.
there's a lot of that, but that's just some research.
Yeah,
Uh,
exactly, exactly. And hopefully maybe even there's a simpler solution. Maybe you won't even have to do the analysis. You can just like find other data and then something else like that. So but yeah, I just want to empower people with knowledge, and I think you're right. I think that if there's tools, then it'll allow people to like arrest that manipulation, because I see it all the time and people come to me, and one of the top three reasons why people come to me is like number three or four is like, hey, I trusted this guy and it's wrong, and now I'm in a huge problem because I thought it was all cool. I just took his word for it, and I don't want anyone to take anyone's word, even my own, for it. That's why I'm always citing sources and stuff.
but to kind of go back to microbial products, it ain't, and
Yes.
also on in defensive, like a lot of products, it's just hard, because shelf stability, you know.
Yeah, or reproducibility. Maybe I mean you could have all the great. My point is that you could have all the great intentions, right? I'm not saying that people are just out there lying all the time, but
Oh
maybe
no,
like, but
microbes behave different in secondary systems, say like I'm using
exactly.
something out in the field compared to like a 5 gallon pot, that's a
Yeah,
whole
yeah.
different world. That's a different
Or
ballgame.
like, what did you do to your plant system that they didn't do to theirs? And it's innocuous, but maybe in this particular case, it's not. So it's just like it just allows everyone on the board to have a more even playing field.
Not saying microbes are bad. Microbes are essential.
Microbes are essential, and I also think that much like the resistance pyramiding in plants and like combining different traits to make them really good and strong and robust and less breakable. The same exact thing can be said for the microbes.
Definitely agree with that, man. Speaking of beneficial microbes, so some of these, I kind of missed out on a. I was dealing with my cat, so I didn't really get to listen to the caterpillar
Oh
slides too
yeah,
much.
yeah, that's fine.
So the caterpillars were taking like genes from beneficial microbes of the plant, or is it
Oh,
sort of
they were just
soil? Or?
taking, so they were taking microbes. I have a couple of slides that explain it in more detail, but like basically, it seems like in budworms and in like fall armyworm and a bunch of other caterpillars, they're really good at manipulating the plant. They do this with their own genes endogenously to them, but they also make use of the genes of the microbes in their gut. And it seems like a lot of caterpillars and budworms in particular, where they grow, where they live, they will like different plants and also different regions. They get different microbes in their gut, but they co-opt a lot of microbes that are even good for the plant. Okay, but then those microbes, they're mercenaries basically. They're just gonna take in input and poop out output, and it doesn't really matter if they're on the plant or in the plant or in the gut of a caterpillar, which might be warm and safe and nice. Um, and so these bacteria and other microbes, they have these capabilities, and they will break down plant defenses for the caterpillar. They will break down the indigestible parts of the caterpillar's diet, and things like this. And they'll even like protect it against like temperature changes and parasitoid wasp larvae that they inject into the body, and even like Beauveria bassiana and fungal pathogens and things like that. So the microbes have a huge effect on the survivability of the caterpillar, and that comes from the soil as well. So soil microbes often are, they often are a source for phylosphere microbes, and then those then become selected by the plant, and then the caterpillar then does its own kind of selection. And then when it feeds on the plant, in the cucumber example, in the wild type, the plant had a defense response that was primed by the microbes. So the plant had a response, it primed the microbes, the microbes reprimed the plant and extended its effect and made it defend against the caterpillar. At least one. When there was multiple ones, it was less effective or not effective at all. In the domesticated one, this effect was the opposite, and the soil microbes didn't matter. They had the wrong response, or they had the opposite effect, and the caterpillar had more capabilities. So it's this three-way handshake of genes, microbes, um, yeah, plant genes, microbes, and then the caterpillar.
Seems like we got a lot of R&D, and I would like more sequencing to happen for sure, so we can just open like GenBank or have it on blast and make data accessibility that way. But like, we just don't know, you know.
We just don't
We
know.
just don't know. And
Yeah,
do I think we'll ever know? Do I think we'll know everything? No, just it's always changing. My my concerns, it seems like shit's changing like quicker than usual. Like you're seeing just like resistant genes or like stuff adapting like real quick, or
yeah,
sometimes vice versa. Um, for example, uh, bud muffs. It's just like that's the thing that scares me so much because like you, they just don't want to die.
yeah. They don't seem like the go out into the dark quietly types. And they're everywhere. They cause, in my budworm video is titled something like 7.8 billion dollar, $78 billion catastrophe or something, because that's the like total agricultural global value annually, more or less. At least in the paper when it was quoted, it's probably more now. Because of inflation. But
Yeah.
it's a lot, and this pest is not just a cannabis pest. So there is research being done about it, but I think but cannabis is really specific in what it needs. So
The corn borer is a big one out here too.
yeah, I've heard people have sent me. That's one of the ones that did line up with my experiences. I have had people talk to me about the corn borer. and the stalk borer too.
I got a question about Florida. So, like, when the moths indoors, is that just, are they sneaky, bad cultivation practices,
Yeah,
or?
I'm not sure. I'm really not sure. It must be. It must be people letting them in. And some of those moths were kind of smaller, but not all of them. The Helicoverpa, as you saw in maybe you didn't see it specifically, but the picture, like the moths are pretty chunky.
Yeah.
It's very they're a very noticeable, like this large animal, a wingspan, and I would use it for bait. Like it's a chunky moth. But the small ones, like the hemp borer, and maybe some of these other ones, maybe they're able to flit around, and if they just deposit their egg, if they happen to be gravid, deposit their eggs and die right after, then that's all you need for the larvae to get in there.
For sure, for sure. I was thinking about that. And another thing on that slide is, like, in addition to moths, was I'm not entomologist by any means, but the Hymenoptera, the wasp and
Yeah,
ant. So,
yeah.
is there
So
any paras? Is there anything that predates on cannabis or parasitizes it,
yeah,
or affect, I should say?
not super much. So yeah, so the Hymenoptera are wasps, and then ants are wasps that evolved to not have wings except when they reproduce, which is why they have wings when they reproduce. It's a ancestral trait. And bees are wasps too, basically.
Hmm.
So that's why I say it that way. And no, not really, to my knowledge. The most pestiferous group in the Hymenoptera for cannabis I've come across are usually ants. Because there's many of them, and so some ants, like red imported fire ants, and some other ants too, like carpenter ants, will attack the stalks. They might eat out the root system sometimes, or they might try to squeeze water out of the tissues. I've seen that in sort of very dry climates.
Really?
Yeah, yeah.
Okay, I can definitely see the ants. They're everywhere. And I got a question that crossed my mind earlier. Seems like a lot of, like, these insects and microbes really prefer varieties that produce cannabinoids instead of grain and fiber. So I was just, as we mentioned earlier, maybe it's like lignin or what have you. But even like some of the grain varieties, I've noticed like there wasn't as much like um it seemed
Like
like
which
they weren't
one?
as susceptible as like our cannabinoid varieties. So
Yeah, it's funny because like I would assume that like the oil that like oil seed, for example, specifically, might be more susceptible to certain pathogens because of all those fats and oils that are nutrient dense. So I could see that being a potential limiter for some of these necrotrophic, like fusarium and botrytis. I wonder though if with the cannabinoid ones, certain cannabinoids are certainly antimicrobial or have other effects, but I wonder if it's just the domestication of cannabis and making the buds chunkier and chunkier and chunkier and frostier, and more moisture locked into the bud inflorescence and things like this, because there's research that shows bud rot goes up with the density of the inflorescence in their study, at least. And I wouldn't be surprised. It's
oh
like two
yeah,
times the density, two times the pathogen likelihood
those
of.
big chunky colas are all nice and pretty to look at, but you'll get some nasty surprises.
I mean, like, look at other crops. I mean, I make this. It's a clarion call for me, because, like, look at tomatoes, look at bananas, look at just about every fruit you eat. It didn't look like that naturally. Corn, like, so we made the parts that we like bigger. It's also the parts that the bugs like, and originally the plant had the defenses to eke out an existence on the smaller fruit, not produce as much nutrients, and maybe produce a lot more toxins we don't like, but we've gotten rid of some of those things, and so then we go, oh, the plant is unhealthy. Oh, it's like, no, I mean, maybe you could make the argument, but not without a lot of help. You can call a fish, you can call a fish a bird, it won't fly. You know what I'm saying? So
Yeah, man, you gave me a lot to think about, and it's all so complex. Even like, because like I'm doing some stuff with like, or I'm part of a group doing stuff with like a soil from various commodity crops and hemp being one, fiber hemp being one of them, and we're looking for nitrogenase genes for nitrogen fixing bacteria. You know, where they pull nitrogen out of you, turn it to nitrate for like the plant.
Mm.
We're looking at that in the soil between various crops to see like do like a real time PCR to see like how's it expressed. But I was also wanting to see if there's a way like we can take like functional genes like that and correlate it with the metagenomic data. Like, oh hey, this group's known to blah 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 that and that, and maybe
Mm.
somehow correlate that over to like the plant. Or it's just
Yeah, that's
it's
the
so
grail, right?
complex, man.
But that's the holy grail. That's what you would want. Yeah, like get some like transcriptomic data, you know, see like over time too, right? Like I have some other slides that go over like, plant defense and resistance and how does that develop and what are the different elements. And, there's so many factors, right? And the big one that often gets omitted is like maturity or time or senescence, like
Oh yeah.
it sucks, but like a lot of these gene expressions aren't going to happen when they're very young. It's going to be different when they're very old, and we're going to harvest them. So of course they're more susceptible to certain things.
I mean, that's why we went throughout the stages of like the crop's life cycle, like grab different soil samples so we can kind of like, oh hey, I mean, assuming like obviously during vegetative stage is when they're probably most active for nitrogenase, just because you need nitrogen. But
Right.
stuff like that. But I would like really like to do stuff like that with like microbiomes and gene expression with like different insects. But it seems like whenever we find like one gene, there's like another gene, and it's just
Yeah.
real hard to keep track of everything. So for like when you're consulting, like how would like you go about say like a client's having a problem? Like how would you go about doing it if like so like you notice these so obviously like doing your research and paying attention to trends, have you noticed like the field that you're in specifically with clients it's just changing really rapidly, and if so, how are you go about to kind of keep ahead of that or keep even with it?
I would say that like because there's an international element to this, it's very variable, and even in the USA, every state's a little bit different. Um, to be honest, I actually feel like it's a lot of influences that are not really directly cultivation specific are causing the most influences, which is sort of a cop out answer, but it's very true. And I wish actually it wasn't true because it would imply that there were more things that we could do about it or that we'd have more of that support we were talking about. But the biggest ones are regulatory, I would say.
Oh yeah,
Um,
the
and
regs.
then, yeah, I mean, what a,
That's the biggest barrier, right?
obviously, right? Yeah. So I mean, but like either people sometimes it's because what's changing is usually that and so like some people in some places they have to like make a plan and then they have to like stay with that plan and they can't deviate from it at all. New product comes out, can't use it, we didn't allow for it. How can you run a business this way? It's untenable. um, and that's just from a commercial angle. If we're just talking about home grow and that sort of a thing too, um, I just feel like the overarching community would be maybe with Hoplite and Viroid, for example, being probably the biggest example. If we had had the support and if the community had been more informed, then we might have had different change. We might have seen different changes. But now people have to make concessions on the onset. And maybe you could argue that a really strategic person would always be making these concessions and have already considered this biosecurity. Okay, sure. But not everyone has the same, you know, logistical constraints. And so I just feel like people like, if they want to defend themselves, they have to make a bunch of concessions about how they grow. So maybe they want to grow outside, could be cheaper. Now they have to do an indoor facility. And oh, well, now that you've done that, or an indoor space or a greenhouse, or they have to build it themselves. However, on the echelon of commerciality to private, we go. But now you have to do all this other um, sort of like buttressing instead of just a more normal, maybe more simple approach because things have advanced so much with certain pests being around and also restrictions that you don't want to get caught with. So
what are your views on pesticides, synthetics versus organic, and even like beneficial insects? You think it's like a combination of like all three, or
What
just
is?
depend? Oh, like as far as like kind of controlling pests and like biological, like say like organic pesticides, synthetic pesticides, or beneficial insects, like a combination of three, depending like what the.
Like, do I recommend that?
Yeah, yeah, yeah.
Hmm. I think that I'm a huge advocate for biocontrols, but I will be the first to say that they're not always appropriate and they're not always the most effective solution. And they are often a very expensive solution, you know, and so, but not always. But I do recommend using biocontrols, certain ones. One really safe example for a lot of people would probably be like Steinernema feltiae or SF nematodes. Um, a lot of people deal with fungus gnats in propagation or in their grow or whatever. And I find SF nematodes pretty much brand agnostically to be a really good purchase. I really like to use them. You can refrigerate them. You can keep them in cold storage, unlike other biocontrols where you have to use them immediately. And if you've never heard that before and you've used biocontrols, that might be why they didn't work for you because they all died. So there's all these constraints with how you use them. But to give you a positive example, and I'll get to your other points about pesticides and the use of organics too. I feel like predatory mites I really like to use in a lot of cases proactively, especially in veg. There's some places where they wouldn't be appropriate because it wouldn't be GACP compliant or whatever, which is fine. But in places where you don't have to worry about that, these mites are really great in veg, especially. They'll feed on like thrips that might come in. They'll feed on russet mites. They'll feed on some other pests that are really odious. And so if you just keep their population levels up, and like if you're a nursery, for example, and you don't even have flowering plants, you don't have to worry about anything else, and you can just keep them on this rotation. So that would be an example, but it has to make sense for what you're trying to do. As far as like organic pesticides, or synthetics, I don't think people should be using if it's systemic and it's a synthetic and it gets in your plant and it stays there forever and whatever. I've had people call me, they'll do this dance where they're like, yeah, I know I'm not supposed to use this, but if I use like a little bit of it and then I take a cutting of 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 a cutting, there's not that much, right? So it's okay. They want me to write off on it for them, and I never do because that's not appropriate. But like that's the kind of stress people are under. They might not even understand. Sort of a tearful example: a friend of mine, very dear friend of mine, he introduced me to his uncle one time, and he's like, "Yeah, this is Matt. He does the pest stuff." He's like, "Oh, Matt, I don't have pests. I just use carbamate." Carbamate, you know, um, very very toxic, very very disgustingly systemic, gets into your plant, will stay there forever. You just dumped it. He died a few years after I met him.
Jeez,
He had other health complications, I'll admit,
is that in the
but
same class of pesticides as like carbofuran and stuff?
yeah, yeah,
Ooh, yeah, it's
yeah. Don't
nasty.
put that on your sandwich. And what I was trying to say earlier with the budworms is that like places in China, in Africa, in India, and the USA sometimes. But like some of the biggest places are like China and parts of Africa, and South America, where illegal use of these noxious pesticides, DDT, organophosphate, carbamate, all these other ones, certain neonicotinoids, they're all creating a huge gym with the budworms, and they're pumping out iron, and they're living in these places where they get sprayed all the time. They're building up all these resistances, and then they're going to export that stuff. So even if you grow really nicely, even if you do the right thing that you're supposed to do, you're going to come under assault. We've outsourced these negative externalities, but now they're coming home to roost, literally, in our fields. So, I feel like the average person has some sort of a voice, and maybe they can hopefully we can all together make long-term decisions that might mitigate this as much as possible moving forward. But but you know, just people say pesticide, they think, oh, bad thing, always a bad thing. Pesticide, no pesticides. But there's like chemical agents and proteins from plants and microbes and other organisms that you can use, like pyrethrin,
Mm.
not permethrin, not pyrethroids. Those are different. But like pyrethrin is from chrysanthemum originally. It degrades rapidly in the presence of light and water, which is abundant in cultivation. Only problem is that if you have a cat, don't be careful when you spray it, because cats lack an enzyme that pretty much all other mammals have that allow them to neutralize it quickly. So it is toxic to cats. But it will degrade very quickly anyways, and then you won't have to worry about it. So that's an example of an insecticidal pesticide that I have very little problem with, especially because it doesn't reside in the plants afterwards. So it's
I
a
never
contrast.
really sprayed. I mean, in the past, I would play with spinosad a little bit, but now it's just beneficials. But I haven't used like mites or nematodes, which I probably should. And it goes like I'll probably ask you a question on nematodes here in a second. The freaking root knot nematodes.
Oh yeah, Melita
Those are kind of
Guyane.
pain in the ass. Um, but oh gosh, now I forgot what I was gonna say. Oh yeah, beneficials. But my plants BT doesn't work anymore. Like on caterpillars, good luck. It's
Really? You're not just
scouting.
saying that to make me feel
No,
validated? Wow.
no, I'm
Okay.
out here in the Midwest. We got super bugs, man. But for me, grasshoppers are the worst. Jeez.
Really, that's interesting. That's really interesting.
I got put nets, spanker bats, scarecrows for birds, but grasshoppers are just the
Grasshopper,
worst.
yeah. That's another one. That's another one where, like, yeah, like I hear a lot about it in like places like the Midwest and even certain parts of California where I'm from. I'm in San Diego myself, but like, like certain parts of California certainly get their share of like large grasshoppers. And I had heard I worked with a nursery for a little while, and they this one in California at least they had like a huge grasshopper problem, and they were out in like Ramona or something. So like the desert, basically.
Oh, you forget. There's those giant locusts out there, ain't there?
Yeah,
I'm not an
I mean,
entomologist, but I thought
there's
they had
some
like.
there's some there's some like large like short horned grasshoppers or like bird grasshoppers they call them because they look like a bird, you know. So yeah,
Man, like I go out there and I'm missing like branches, and I see just a
yeah.
grasshopper sitting there like munching on it. So like, just putting up scarecrows, even like nets. But
Net.
grasshoppers for me were like the worst, though.
So what? So did you feel like the nets were really effective? Because that kind of physical barrier, if you do it right, I feel like is like, you know, if
Moths
it's a huge.
like to still get in though, like with their eggs and stuff.
Yeah, you need like mesh screen. But I
Yeah,
didn't mention this about the budworms, but like one of the because I mostly just focused on why are these things a huge issue. But like, if you don't let the moth lay the egg in the first place, then you're fine. Because the caterpillars are not like army worms that like move across the ground to find another host usually. So that's all you need to do. And it's a bit of an investment, but it's so much worth it, especially if you don't have a huge field. If you have a huge field, that might not be tenable, but if it is tenable, I recommend that thrips screen for thrips and spider mites and rusted mites that can travel on the air, believe it or not, and things like this, and then maybe something else outside of that or inside of it.
but yet our fiber field was fine, and we had 70 acres, no insects or anything. Just literally,
Really?
yeah, literally just planted it
Uh,
on cow manure, and it just woof. There's actually an old video on FCP of us harvesting it, and I'm walking through the field, but no, nothing at all. Like no infection, like nothing, nothing, nothing. No
Nothing.
PM, no botrytis. Well, I mean it's by, I mean hardly in vegetation, so you're not gonna get like botrytis stuff, but like no insect damage, no pathogens, like nothing. It was healthy as could be. So like I think there's something there. Is it just because like these
I think
varieties
there
just
is.
used for forever? I mean, because I mean there's uniform as could be like when you grow them. There's like no variation. They're all very
I think
uniform.
I think there is something
So
to it. I really do. And actually, I don't have nearly as much hemp experience as I wish that I did, to be honest. It's not saying none, but I'm really interested in like seeing livestock feed get more of a comeuppance. I'm seeing comeuppance, excuse me. But I want to see it actually come out and be viable. And I also want to see like oilseed and food too as well.
Oh
But
yeah.
uh, but I do believe you're right. And how much of it is like maybe like like selection from those pressures? Maybe that's part of it. More abstractly and directly, I think it is. But I think more directly, I think you're right about the lignin idea, and like maybe also, I think there's also just something to be said that like a lot of pests are feeding on a lot of like bud associated pests, especially they need that flower material. So if you don't have that, then that's going to really cut it down, perhaps.
Okay, that definitely makes sense. It'd definitely be interesting to look at because like even some of the grain varieties I've been around, like there wasn't too much happening with them.
The Florida paper had mentioned something about that too. So like, what were they saying? Like they're saying some pests, they were an issue because they fed mostly on the seeds, and certainly there are like Lygus likes to feed on the fruiting bodies of plants, even if that's like the seed, the achene, right? But so like you might get some like seed bugs, and maybe if you grow only seed hemp, or you're exclusively growing for seed for breeding or for just industrial reasons or production or whatever, um like grain, sorry, excuse me. But that might be an issue that some people get like primarily, and it's just going to be unique to them.
Man, so many directions we can go.
Yes, there is. I don't have any particular bend.
So are you doing some current like research right now too? Because I know you're going to Europe. Is there anything you're doing over in Europe by any chance?
Yeah, so I'll
Research?
be so very soon I'll be attending a an event in the Netherlands. Inexo's, let's just spell it INNEXO. Inexo has a acceleration day in the Netherlands, and they're doing some research. They're looking at, if you can believe it, they apparently have a model that works, that is able to tell. And if you had told me this like even as much as 5 years ago, I would have said that I would be curious, optimistic, but ultimately I didn't think it would be possible. But apparently, he's able to, just through visual scanning of seeds, tell whether they're male or female, and used a really robust like machine learning system to tell the difference. And apparently, he's done a lot of trials. That was a really cool example, but more cultivation related.
Like something with like the pericarp way it's shaped, or can you like spectrum?
Something about believe it or not, it's like what people were saying a long time ago, like coloration. It's not the same things that people were saying. Don't get me wrong, and I don't think that it is. What do I want to say? I don't think that is so granular that a person could just eyeball it and have like close to 50% even of the ability. So it does seem to be very unique, however this process goes. But I don't want to speak too much about it. I'm somewhat ignorant about all the details, but
Sounds like something you can use in the field, or like
yeah.
a,
Oh, or what?
or just like any cultivator, seed producer.
Yeah, or like yeah, that's kind of I think the idea there. But no, he's also working on like no veg. Have you heard of this?
No veg, like no. I assume you just flip the flower immediately, right?
That's right. Basically, Dom Van Drusen, you guys can look him up. He is working on like plants, and he's showing that plants they don't have a detriment, like, because that's what people will say, like, oh, if you just start from flower, no veg time, the plant's not going to be stronger. The plant's going to have not as much yield or biomass or whatever. But according to his trials, and he has his own R&D space where he does this research, he's been able to show that there's plants that are not detrimented at all, or even excel from no period of vegetation, and it's quicker. Um, so there seems to be some validity to that.
Really? Because I mean, if you look at the ornamental industry, they're not growing big plants; they're growing small, squat plants, and they can get more like yield per square foot. I mean, like um,
Yeah,
for example, the gosh, I'm having a brain fart. The poinsettias, boom, squat.
yeah. Do you want to know a funny story that I have about poinsettias?
Yeah,
I used to work with a cut flower company that put poinsettias on the map. And they were able to do that because up until when they started, poinsettias were a single stalk plant, all the cultivars. But they were able to find one that's multi stalk and they could take cuttings. Now, they thought it was genetic and they patented it, the cultivar. Come to turn to find out it's not genetic. It's because there was a little microbe, a phytoplasma. Usually
Really?
phytoplasmas tend to be pathogenic, or at the very least they're not parasites even necessarily, or they're hosts, in that they are trying to hurt the plant and gather resources aggressively in a damaging way. But they are pathogenic because their presence will cause hormone manipulation changes in the hormone of the plant, which is usually not good. But in this one way, it was good. It caused the plants to be multi-stalk. And because
Hmm.
it's passed down from parent to offspring through vertical transmission, they thought it was inherent to the plant. Isn't that crazy?
I wonder if there's something we can do like that with a cannabis where it's self-topping, like maybe like a microbial like influence.
Maybe. Yeah, possibly. But like what you said about like that domestication phenotype is true. That like it's like Brussels sprouts. That's usually the example I like to give. We don't need any of the other foliage. We just need the particular section, or broccoli is a good example. Probably broccoli is the superior example since that's the floral buds. But yeah, that's all it is. And then the rest of it we don't really want. Um, or you can use it for other things, certainly. And so for cannabis, I think there's a likelihood of that. I heard people talking about that in Israel too, sort of like doing just flowering from the base and like having a bunch of like Brussels sprouts esque cannabis stalks, which just like one big nug. And then you just harvest the one big nug, and then it's like easier and that kind of a thing. I don't know how far that research has gone, but yeah, I think that there's something to it. And I think he's also looked at under canopy lighting as well, and I think he's a proponent too. I hear this also in the gestalt of cannabis, and it's not something I've done a lot of myself personal looking into, but it makes sense to me that it would be good. The only issues I had, and it seems like Dom has some changes or has some ideas about how he's implemented it to assuage those concerns, which is that if you have lights at the top at the bottom, it might be really hard to like treat the plants. Like if you have pests, for example. But apparently, he was able to develop a system where you can, get them out of the way or whatever. So that's easy enough. But yeah, what do you think about that?
All right, so under canopy lighting and over canopy lighting make it difficult to kind of like get to the treat the plant for pests. Because I'm like when you say something like that, I'm thinking like a
Yeah.
tier system
Yeah, yeah,
almost.
yeah, yeah. I guess I don't know exactly the structure, so I don't want to misspeak. But it seemed like the plant grew up, out, and then you had lights here, and you had like lights beaming up. So you get a more total. And like I think he took off some of the lighting at the top and made it more sort of equivocal, you
Mm.
know, across it, and so the plant grew out with alacrity.
Oh, okay. So like side lighting.
Is
If
that not
I'm
the same?
understanding,
Maybe I'm speaking wrong, but yeah, like
so like
yeah, under
lights surrounding
canopy.
the.
Well, I don't no, he was doing like under canopy, but like the idea is that he would get coverage.
Oh yeah.
Maybe not in the same way that like a whole wheel would, but yeah,
No, I'm a proponent of under canopy lighting.
yeah,
It's getting
yeah.
rid
I
of
hear a lot of
LARF.
people talking
LARF's
about
not
it.
a thing anymore.
Right, right. So I think there's benefits to that.
It's expensive though.
Yeah, so that's the thing that keeps being a logistical component that some people will be able to use and some people will not be able to use, right?
That's
And
why
is
I
it
was
worth
kind
the
of.
investment?
That's why I was kind of thinking like flipping right away because like you look at ornamentals, it's just small squat plants, and plus think about you can like if they're in flower they're gonna finish it, and you just rotate them really quick, instead of
Yeah,
like growing it out with a certain type and doing all that stuff, putting up trellis and the cut down on labor costs. Now the quality of flower, I don't know. I never smoked anything straight from like that's been flipped right away. I don't know about the quality, but just like based on what I've seen with like other commodities, especially in ornamentals, it's just small squat plants, and I can see
yeah.
I can see like the cannabis industry going towards that indoor model. Me, I tend to be more outdoor. I have no problem with indoor because I mean, they said live out here like in the Midwest, some really good soil. I mean, the selection pressures with the insects, that's just a motherfucker of it. But other
But
than that,
that's the thing. But that's the thing, right? It's like you can have 100% of nothing, or like, you have 10% left, and that's great. But people can't afford that logistical cost. It's just too much. And it's not like as easy as like, buy this product and spray it, right? For one thing, regulations, but for another thing, people don't want to. People don't get into growing, as I've become very commonly saying. People don't go into growing to catch them all and create your Pokedex of
yeah,
pests. That's not what people got into growing in. They got into growing so they could grow plants. Maybe they like growing plants. Maybe they like the resources they make. Whatever. That's not what people want. So yeah. Whatever they can buy to like not have that problem is a huge boon, but it might be expensive or make the job way too complicated.
man, I wish I can talk more and more, but I got some chores to run. But man, feel free to come on anytime you want. And who knows? I might even hit you
Absolutely.
up, just have a conversation like behind the scenes because I can bounce
That's
ideas
fine.
back and forth with so much. But
Did we answer all the
um,
questions?
oh, see,
It's okay with me.
yeah, I think I don't see really anything here in chat that popped up.
Well, that's good.
I mean, I got more, but dude, we can save that for another day.
Let's save it for another day. Yeah, do your chores. Have a good one. This is really fun for me, at least. And yeah, I love answering questions. We can do another one of these in the future. I'll probably be available in the near future for that. Yeah. And yeah, just contact me anytime, James. This is a pleasant conversation.
I would definitely be interested to hear about your Netherlands trip. That sounds like there's some interesting stuff.
The what?
Oh, your European trip sounds like you're going to be touching upon some interesting stuff. I'm especially
Yeah,
interested with like the flipping straight from
from
you know,
Flower, the no veg.
yeah, the
Yeah,
no
look
veg.
him up. Looking up Dominic with a QUE at the end because he's from Austria. Dominic von Grusen. Yeah, he's doing all that research. Check him out. I think he's really insightful. I think he's got some cool ideas.
So you're going to the conference out in the Netherlands. There was that group. I cannot pronounce the name of the university, but they're the ones that reconstructed those ancestral like synthesis.
Yes, I saw that research.
That was insane. I was like,
That was really
whoa,
cool.
that was some next level stuff. But yeah, man, I would definitely love to pick your mind again. Your presentation brought up like a million other questions in my mind. So how do we yeah, we'll save for another day.
I'll try to only have a million answers.
A lot of it's about microbiomes and potential horizontal gene transfer between microbes and
Oh yeah.
all that
And
fun
plants
stuff.
and pests. Yeah, I think there's some cool stuff there, and who knows what us as humans are doing too. Positive, negative. We'll talk about that later.
Oh yeah. All right, Matthew, thank you so much. And to my class, I'll have those essays graded by tomorrow. So far, y'all doing great. Keep up the good work, and I will be bothering you here tomorrow. All right, all right. Peace out, y'all.
Have a good one.